Hello everyone, welcome back to my YouTube channel Microbe Academy. We are on the microbiology lecture series of types of Plasmodium, and today we are going to learn about the Plasmodium falciparum. The name falciparum comes from the characteristic sickle shape of the gametocytes of this species, and it is highly pathogenic of all the Plasmodium species. And by the end of this lecture, you will be able to describe the life cycle, morphology, pathogenesis, and clinical significance of the Plasmodium falciparum. Okay. Why the falciparum matters more than the other species of the Plasmodium? The answer is this is the highly pathogenic of all the Plasmodium species, and the disease has a high rate of complications and unless treated is often fatal, and the species is responsible for almost all the death caused by the malaria worldwide. And the extremely high because of the high rate of the complications and fatality, this parasite is considered as the mm, key point of the matters among the other parasites. The parasite belongs to the kingdom Apicomplexae, and the vector is same as the female Anopheles mosquito, and the four red blood cells of the all ages are infected by the parasites, and fever periodicity is in every 36 to 48 hours and is the only species with no hypnozoites. Okay, now let's learn about the different phases of the parasites. During the pre-erythrocytic phase or the schizogony, the sporozoites of the parasites are sickle shape, and the tissue phase consists of only a single cycle of the pre-erythrocytic schizogony. There are no hypnozoites of presence, and the matured liver season can release about the 30,000 of the merozoites. Okay, now let's learn about the ring forms and trophozoites of the Plasmodium falciparum. The early ring form in the erythrocyte is very delicate and tiny, measuring only one-sixth of the RBC diameter, and ring are often seen attached along the margin of the red blood cells, so-called form a call, and binucleate ring, common resembling the stereo headphones in appearance, and several rings may be seen within a single erythrocyte. In course of the time, the ring become larger, about one third of the size of the RBC, and have 1 to 2 grains of the pigment in the cytoplasm, which we can also see in the later figure. The subsequent stages of the asexual cycle, the late trophozoite, early and the mature seasons, are not ordinarily seen in the peripheral blood smear of the Plasmodium falciparum, except in a very severe or pernicious malaria condition, and the presence of a falciparum seasoned in the peripheral smears indicates a grave prognosis of the disease and need a prompt medical treatment. The mature season of the parasite is smaller than in any other species and has 8 to 24, usually 16 merozoites. In Plasmodium falciparum, the erythrocytic schizogony takes about 48 hours or less, so that the periodicity of the febrile paroxysm is 36 to 48 hours. Okay, now let's discuss what changes arises in the infected red blood cells. The infected red blood cells are of a normal size, unlike the Plasmodium vivax, which has a enlarged RBC and show a brassy coloration around the microscopy. Both young and mature RBC are attacked. Because of this, there is a large parasitized population, and there is presence of a 6 to 12 coarse brick red dot visible inside the infected RBC. These are known as the Murray's clefts, which is the diagnostic feature of the parasite, and it represents the protein export machinery of the parasite. Some infected RBC shows the basophilic stippling. And talking about the parasitization rate, Yeah, the Plasmodium falciparum uniquely has a high intensity as compared to the other species. Up to the 50% of the all RBC can be infected in a very severe cases, and this massive parasitemia derives the multi-organ failure and explains the high mortality rate of the parasite if untreated. Okay, now let's learn about the gametogony stage of the parasites. These are also known as the diagnostic crescents of the Plasmodium falciparum. The gametocide cycle starts after the several rounds of the schizogony, and the gametocytes appear in the circulations in 10 days after the ring stage, and early gametocytes are rarely seen in the peripheral blood, but the mature gametocytes are the diagnostic stage present in the blood smear. Mature gametocytes, which are seen in the peripheral smears, are curved oblong structures described as a crescentic sickle sausage or banana shaped, and they are usually referred to as a crescents and can survive in a circulation for up to the 60 days. The comparison between the male gametocytes and female gametocytes, the shape of the male microgametocyte is broad, sausage, kidney shaped as compared to the female gametocyte, which is thin and typically crescentic. And the end of the microgametocyte is blunt, rounded, and as compared to the female macrogametocyte, which is sharply rounded or with a pointed end. The cytoplasm is pale blue or pink color in microgametocytes, whereas it is deep blue color in a macrogametocyte. And the nucleus of the microgametocyte is pink and large, diffuse, but the nucleus of female macrogametocyte is deep red and compact in structures. And the pigment structure is scattered in cytoplasm of the microgametocyte, whereas in macrogametocyte the pigment is clustered around the nucleus. Here in the figure we can see the female gametocytes crescent with a central nucleus and a male gametocytes in the figure of the Plasmodium falciparum. And the infected erythrocytes, there is a marginal ring form, and the young ring stays in the ring form with a double chromatin dots at the mature ring form. And the mature ring forms has a particular mirrors dots, and the trophozoite is an amoeboid stage containing the chromatin division, and the nuclear divisions and development of the merozoites can be seen in the figure. And the mature seasons with a centralized pigment is present in the and with advanced merozoite development, but this advanced merozoite development with commencing pigmentations and seasoned mature with the centralized pigments are rarely seen in the peripheral circulation of the Plasmodium falciparum. This is the general overview of the several morphological features seen in the smear of the Plasmodium falciparum by the Giemsa stains under the high magnification power. Infection due to the Plasmodium falciparum is responsible for the cause of the malignant malaria or a pernicious malaria. And the pathogenesis of the malignant malaria involved the nerve formation, cytoadherence, and rosetting and capillary plugging, leading to the cerebral malaria and multi-organ failures. And the pathogenesis involved during the late stage seasons of Plasmodium falciparum secretes the protein on the surface of the RBC to form a knob-like protuberance in a erythrocyte cell membrane, and these knobs produce the specific adhesive Plasmodium falciparum erythrocyte membrane protein 1, so that the infected RBC become the sticky, and this Plasmodium falciparum erythrocyte membrane protein 1 can bind to the endothelial receptors, and infected RBC can also bind the uninfected RBC, forming the rosettes, and the interferon gamma and other cytokines can upregulate these receptors, and This cytoadherence and rosetting lead to the capillary blockage in the brain, kidney, lung, placenta, spleen, intestine, and bone marrow, leading to the cerebral malaria and multi-organ failure. And similarly, other virulence factors such as histidine-rich protein, which is also used in the rapid diagnostic test, is also responsible for the pathogenesis of malignant malaria caused by the Plasmodium falciparum species. Okay, now let's just quickly summarize the whole topic. The name is arise from the sickle cell gametocyte, which is also a diagnostic feature of the Plasmodium falciparum, and there is only one peri-erythrocytic cycle and no dormant liver stage, so there is no relapse, unlike in the vivax and ovale. The smallest ring stage is it presents with a binucleated ring or a multiple ring per cells is present, and the infected RBC is normal in size with a brassy color and 6 to 12 mirror scleps and up to with a 50% of the parasitemia. And the presence of a seizant in the peripheral blood smear indicate the grave prognosis and severe malaria emergency. And the gametocytes of the Plasmodium falciparum has a characteristic crescentic or a banana shape, where the microgametocyte and macrogametocytes has a different color under the staining. And the pathogenesis involve the formation of nerves and cytoadherence and rosetting, leading to the capillary clogging and cerebral malaria and multiple organ failures. And the several virulence factors such as PFEM1, HRP, histidine-rich protein, etc., are responsible to trigger the inflammations and causing the severe malarial infections in Plasmodium falciparum species. Thank you for watching. If you found this video useful, please don't forget to like, share, subscribe and press the bell icon. If you have any queries, please let me know on the comment sections. Till then, see you in the next video on the series of types of malarial parasite. Thank you.